So today we'll perform the gram staining technique. So bacteria are prokaryotic, microscopic, single-celled organisms that thrive in the diverse environment, and it constitutes a large domain of prokaryotic organisms. So these bacteria, they are classified as gram-positive and gram-negative based on the differences in the cell wall structure and their response to gram staining. The gram staining technique was developed by Dr. Hans Christian Gram in the year 1884. In this technique, the Gram reaction, it is related to the permeability of the bacterial cell wall and the cytoplasmic membrane to the diiodine complex. In this process, the fixed bacterial smear is subjected to 4 different reagents, that is crystal violet, which is the primary stain, then iodine solution, which serves as a mordant, then alcohol, which is a decolorizing agent, and finally safranin, which is the counterstain. The color changes that occur in the bacterial cells in each stage is due to these 4 reagents. The bacteria which retains the primary stain, that is crystal violet, and appear dark blue or violet in color are the gram-positive, whereas those that lose crystal violet and counterstain safranin that appear pink to red in color is the gram-negative bacteria. Gram staining, it is based on the ability of the bacterial cell wall to retain the primary stain crystal violet dye during alcohol treatment. The cell wall of gram-positive microorganisms have the higher peptidoglycan and lower lipid content than as compared to the gram-negative bacteria. So iodine is subsequently added as the mordant to form the crystal violet iodine complex so that the dye cannot be removed easily. This step, it fixed the dye to the cell wall. However, subsequent treatment with a decolorizer, which is 95% ethanol, it dissolves the lipid layer present in the gram-negative cells. The removal of the lipid layer, it enhances the leaching of the primary stain, that is crystal violet, from the cells into the surrounding. In contrast, the alcohol dehydrates the thicker gram-positive cell walls, closing the pores as the cell wall shrinks during the dehydration. As a result, the diffusion of the violet iodine complex, that is the crystal violet iodine complex, is blocked and the bacteria remain stained. The length of the decolorization is critical in differentiating the gram-positive bacteria from the gram-negative bacteria. A prolonged exposure to the decolorizing agent, it will remove all the stain from both types of bacteria. Finally, a counterstain safranin, it is applied to the smear to give decolorized gram-negative bacteria a pink color. So now we'll see the requirements. First one is the 24 hours bacterial culture. Then gram staining reagents, which includes the crystal violet, grams iodine solution, 95% ethanol, and safranin. Next is the staining tray, then wash bottles, droppers, inoculation loops, glass slides, spirit lamp, microscope, and so on. So now we'll see the procedure. First, a fresh clean slide is taken and the smear area is marked on the underside of the slide with a marker. Then the slide is flamed and is placed on the working table. Then the tubes containing the culture is rocked for attaining uniform distribution of the microorganism throughout the broth. Inoculation loop is heated till red hot by placing the loop at 60-degree angle into the upper core of the spirit lamp flame by holding it in the right hand. Then the loop is allowed to cool and 1 loopful of the bacterial culture is transferred from the tube to the slide. Further, the mouth of the culture tube is flamed and a cotton plug is replaced again. Next, the inoculation loop is heated again, and when it is cool, the bacterial suspension is spread thinly over an area with the help of the inoculation loop. Finally, the smear is allowed to air dry. So that was about the preparation of the smear of the liquid culture, and now we'll see the solid culture. So how we prepare the smear for solid culture. For solid culture, A loopful of the sterile distilled water is placed on a clean, grease-free glass slide. Then aseptically, a small amount of bacterial colony is transferred into the distilled water droplet with the help of the inoculating loop. Then the culture is emulsified with the water droplet with the loop. Further, gently the slide is heated by holding it in the right hand above the flame. Now we'll come to fixing a smear. How to fix a smear? The smear is passed through the tip of the flame very rapidly for around 4 to 5 times. Then the smear is allowed to cool and air dry. So now we'll come to staining. Further, the slide is placed on the slide rack. Then the smear is flooded with the crystal violet stain and it's allowed to react for 1 minute. The slide is then washed with distilled water for few seconds using the wash bottle. Then the smear is again flooded with grams iodine solution and it's allowed to react for 1 minute. So this addition of grams iodine solution to the smear, it forms the crystal violet iodine complex, and the complex so formed, it prevents the dye from washing away easily. And this step fixed the dye to the cell wall. Next, the iodine solution from the slide is washed up by the use of 95% ethanol. So ethyl alcohol should be added drop by drop until no more color flows from the slide. In this step, the gram-positive bacteria are not affected while all the gram-negative bacteria are completely decolorized. Then the slide is washed with distilled water for few seconds using the wash bottle. Finally, the slide is flooded with counterstained safranin and it's allowed to again react for another 1 minute. Next, the slide is again washed with distilled water for few seconds using the wash bottle. Then the slide is blow dry by the use of absorbent pepper and the stained slide is allowed to air dry. So, under observation, the slide is microscopically observed using the oil immersion objective in the microscope. So, in the result, under the microscope, the given bacteria appear purple in color and are arranged in cluster and hence it is the gram-positive bacteria. So that was about the gram staining technique, and at the end I would like to add that the purpose of the gram staining is to separate the bacteria into two different categories, that is the gram positive bacteria and the gram negative bacteria. So this classification, it is based on the chemical and the physical properties of their cell wall constituents. The result of the gram staining reaction reveals whether the organism is positive or negative based on the color the organism appears after staining. So gram-positive organisms will not be easily decolorized and retain the purple stain of crystal violet, while gram-negative organisms will be decolorized by the alcohol and are subsequently stained by the safranin and it appears red or pink in color. So today we'll measure the bacterial growth in the broth. So we all know that the ultimate goal of any species is to make more of its own kind. This fundamental law, it ensures survival of the species. An increase in cellular constituent is termed as the growth. A typical bacterium, it grows by increasing its length and masses, which facilitates the expansion of its nucleate as its DNA replicates. As DNA replication comes near to completion, the cell, in response to complex genetic signals, begins to synthesize an equatorial septum that ultimately separates the two daughter cells. Such a process is termed as the binary fission. In this process, one parent cell, it splits into two daughter cells. Usually, microorganisms, they are cultivated in the liquid medium and are grown in the batch culture or the closed system. So as no fresh medium is provided during incubation, nutrient concentration, it gets declined and the concentration of the waste increases. Further, the growth of the microorganism can be plotted as the logarithm of the cell number versus the incubation time. The resulting curve, it has 4 distinct phases. The first phase is the lag phase. Second one is the log, or also known as exponential phase. The third one is the stationary phase. And finally, it is the death or the decline phase. So first we'll come to the lag phase. During this period, that is the lag phase, there is no immediate increase in cell number or mass when microbes are introduced into the fresh medium, and therefore this period is called the lag phase. During lag phase, the cell it synthesizes new components, although cell division does not take place. Lag phase it varies considerably in length with the condition of the microorganisms and the nature of the medium. These phases may be quite long if the inoculum is from an old culture or one that has been refrigerated. So now we'll see the exponential phase. Microorganism, it grows and divides at the maximal rate during the exponential or the log phase. So their rate of growth is constant during the exponential phase. That is, the microorganisms are dividing and doubling in number at regular intervals. As each individual divides at a slightly different moment, the growth curve it rises smoothly. Then the population of microorganisms is considered to be most uniform in terms of chemical and physiological properties during these phases, and hence exponential phase culture are usually used in biochemical and physiological studies. So in the stationary phase, microbial population growth ceases and the growth curve it becomes horizontal during a phase that is known as stationary phase. During this phase, that is the stationary phase, bacteria attain a population level of around 10 to the power 9 cells per ml. But the final population size it depends on nutrient availability and other factors such as the type of microorganism being cultured. During this phase, the total number of viable microorganisms it remains constant. This may result from a balance between the cell division and the cell death, or the population may simply cease to divide though they remain metabolically active. So microbial population they enter the stationary phase due to several factors. The first factor is the nutrient limitation. The second one is limited oxygen availability. And the third one is the accumulation of toxic waste product. The final phase is the death phase. So various detrimental environmental changes such as nutrient deprivation and the accumulation of toxic waste, it leads to the decline in the number of viable cells. Such a phase is termed as the decline phase. The death of a microbial population is like its growth during the exponential phase, which is usually logarithmic. A constant proportion of cells it dies every hour. Although most of the microbial population it dies logarithmically, the death rate may decrease after the population has been drastically reduced. So this is due to the extended survival period of a particular resistant cell. So now we'll come to principle. Determination of bacterial growth, it requires an inoculation of a sterile broth medium and the incubation of the culture under optimum temperature and gaseous condition. The bacteria they will reproduce rapidly under the given condition, and the microbial growth may be measured by turbidity measurement. So this technique, that is the turbidity measurements, it relates the cell number to the turbidity or the cloudiness of a broth culture. Turbidity or optical density is not the direct measurement of bacterial cell numbers. Optical density is directly proportional to the cell concentration. Here, in our present technique, turbidity is measured by the use of a spectrophotometer. It is an instrument that measures the amount of light that is transmitted directly through a sample. So, with the increase of the bacterial numbers, the broth becomes more turbid and it causes the light to scatter and allow less light to reach the photoelectric cell. So, the change in the direction of light is indicated in the spectrophotometer as the percentage of transmittance. So this percentage of transmittance is the amount of the light passing through the suspension, and the absorbance. So this absorbance is a value that is derived from the percentage of transmission. So the relationship between the percentage transmission and the absorbance is exactly the same as the relationship between percentage transmission and the optical density, that is OD. Thus, we can read the values on the log scale, that is absorbance, and consider this to be the OD value, that is the optical density value. Bacterial growth curve construction, it requires a sample of 24 hours check flux culture. So now we'll come to requirements. So broad culture of bacteria is required. Then sterile 5 ml pipettes, spectrophotometer, cuvettes, spirit lamp, etc. So now we'll come to the procedure. Aseptically, 5 ml of the broad culture is transferred to the 100 ml of the nutrient broth in the flask. Next, the initial OD, that is the initial optical density, is determined at 600 nanometer wavelength. The inoculated flask is kept in the shaker and it is set at 120 rpm at 37 degrees Celsius for 6 hours. Further, aseptically, after 30 minutes of incubation, 5 ml of the culture is transferred to the cuvette. Then, the optical density of the sample is determined at 600 nanometer. The step 4 and 5 at each 30 minutes interval is repeated for a period of 6 hours. So in the results, the optical densities of the broad culture suspension at 30 minutes time interval for 6 hours is recorded. So to conclude, bacteria, like other forms of life, must have proper environmental conditions including temperature, sufficient space and nutrient in order to survive. Given the relative simple nature of the replicative process of bacteria under optimal conditions, their number will increase geometrically in a very short period of time. And enumeration of the microorganism is especially important in dairy microbiology, food microbiology, and water microbiology. Knowing the bacterial count in drinking water, fresh milk, yogurt, etc. can be very useful in many aspects of industrial microbiology.